Hello and welcome everyone to today's webinar that is brought to you by Quanterix in partnership with AZO Network and is titled Steps Forward to Extracellular Vesicles Direct Analysis from Ultrasensitivity to Handcrafted Solutions. A big thank you to everyone involved in making today's event possible and to you in the audience for joining us from across the world. I'd first like to introduce today's fantastic speaker and then explain a little bit about the format for today's event. So I'm delighted to welcome Alessandro Gori. Alessandro holds a PhD in medicinal chemistry from the University of Milan, and he is currently a research scientist and group leader at the National Research Council of Italy. His research is devoted to the development of chemistry-based solutions for applications in biosciences, including the customization of analytical platforms and assays to detect pathology-related biomarkers. Recently, his efforts have been strongly focused on extracellular vesicles as emerging analytes towards liquid biopsy applications. A big welcome to Alessandro today. The webinar will run as two parts. In just a moment, Alessandro will start off with a 40-minute presentation before we move things into an audience Q&A. So if you have any questions at any stage, please write them in the Q&A box, which you'll find on the right-hand side of your screen, and we will answer as many as we can following the presentation. Quick note that if you are watching this back on demand, then you can still ask your questions, but you'll receive a reply via email. Finally, before we get started, just a few notes. The webinar is being recorded. We'll share the link with you within the next 24 hours, so please do share that email with anyone else you feel would benefit from watching today's content. And you can, of course, watch back on demand anything that you missed today. If you have any technical issues, then these are normally resolved by refreshing your browser. If this doesn't work, send us a note in the Q&A box and we'll get you back up and running as quickly as we can. As I mentioned, there will be a Q&A following the presentation, so do send those in at any stage during the webinar today. And if we don't get to them in the Q&A session, we'll follow up via email at a later date. Right, let's get things started with the presentation.
Good morning, everyone, and thank you for this kind introduction, which I'm very, very happy to be here today and to share with you our latest development in the area of extracellular vesicles analysis, and to show you a bit more about what we do every day, and especially I will focus on which is our approach to develop new solutions into the analytical field in general, and how we combine technology and molecular tools to bring new solution. So again, just to give you a very brief overview about who we are a group located in Milano. We call ourselves Chemistry and Technology for Bioscience Group. And within the group, we have many different expertises, ranging from chemistry to biology and technology, and at many different sides. Myself, I'm a chemistry by training, especially I focus most of my career in peptide chemistry under many different angles and different sides, from bioactive compounds to their use of peptides to their integration into microanalytic device, which bring me to today's webinar. Because one of the key components of our lab is also bioassay development, and especially in miniaturized systems. And particularly, we spent a lot of time in recent years to develop tools for the analysis of extracellular vesicles towards their phenotyping and single vesicle analysis toward their prospective use. And pretty much this is what I will focus my presentation today. Again, a brief introduction about what are extracellular vesicles and why they are so relevant in this moment to us and to many people actually. So basically, extracellular vesicles are some biological nanoparticles which are released by every kind of cells, and which are emerging as a very crucial way of cellular communication, intercellular communications. And this is in particular because they basically carry a lot of biomolecular messengers that can be shared between cells. And so this role, basically, they're kind of cellular message in a bottle. And this relevance is increasingly appreciated in both the regulation of physiological processes, but also in pathological conditions and host-patient interaction as well. This is let's say, a prototypical composition of an extracellular vesicles, which basically can include any possible biomolecule, so ranging from lipidic membranes to membrane proteins, internal proteins, enzymes, DNA cargo, RNA cargo, and so on. And so, as you can see here, they're investigated for many different areas and application. So I try to group what I feel are the two main ones. The first one, the key assumption is that the composition of extracellular vesicles is very close to their cell of origin, meaning that if we are able to intercept vesicles which are released in all biofluids, from plasma to urine to CSF, we have a tool to try to decode the composition of the cellular of origin, especially to decode the presence of putative biomarkers which can be related to a pathology. And so this brings to the main area of investigation, which is the one associated to biomarkers discovery, liquid biopsy applications, and so on. The other way around, as basically their intrinsic function in the body is to share information between cells, and they're sort of made by cells for cells nanovectors that can be somehow used in place of synthetic nanodelivery systems, for example, in vaccine delivery or other therapeutic deliveries. They're perceived as more biocompatible somehow, and they can basically improve quality in enhancing, in crossing barriers, and be very selective in the target. But also on the other way, they can be basically used as therapeutic themselves, especially in the area of cell-free therapy, regeneration, and so on. But again, really, the field is very broad, and the applications today are countless. And this is basically to support what I'm saying. So we are kind of living in a period where extracellular vesicles are really gaining the spotlight, and we are experiencing a massive increase in the interest that they're gaining. But the interesting thing here is that they've actually been known for a while. So the first aspect that I want to highlight is why we are today, let's say, why now? And this is pretty much related to technology development, which basically enabled the study of extracellular vesicles. As we will see, they're very complex matter of investigation, despite very interesting. And so just think about the evolution of microscopy techniques that we've been experiencing in the past few years, or many other techniques which are basically enabling us to have a better look of EVs and to understand more and more their roles, and possibly to open the scenario towards their actual use, and both in therapy, diagnostics, or real-world applications. So why we need such a level of technology? Because basically we have a multi-layer complexity when we deal with EVs. The first one is their inherent complexity. Basically they are starting from the point that they're not, let's say, traditional molecules or analytes. They're basically nanoscale or mesoscale particles, which are intrinsically heterogeneous in terms of size. They're also heterogeneous in terms of what they bring in terms of their cargo, surface markers. And so overall, when we talk about extracellular vesicle, we actually really talking about something extremely heterogeneous. And so, given the fact that they're also nanoscale objects, these are already add our first level of complexity to their investigation and study. On the other side, they're not the only circulating bio-nanoparticles, because if you do see under, let's say, a biophysical eye, the whole secretome, basically other components of the human secretome are biological nanoparticles, and these mostly include lipoproteins, because basically they share some properties with EV, including overlapping sites and some features. And the tricky part of it is that lipoproteins and other lipoparticles are actually present in a much more abundant way than EVs. And this needs to be coupled to one thing, that separations at the nanoscale are not trivial at all. So you can use different methods, different approaches to separate the components of the nanostructure secretome. But basically, depending on the method that you choose, they can range from centrifugation, size exclusion chromatography, immunoaffinity separation. Basically, there's no a true optimal way, and somehow the when you choose your separation method that you want to introduce upstream your analysis for whichever reason you are going to analyze your sample, basically you are introducing a trade-off between yield of recovery, purity of what you are getting, specificity versus yields, basically, or versus purity sometimes. And basically, depending on the technique, on the strategy that you want to use for purification, you are introducing a bias, which can be a bias of different contaminants, which you do not separate by that techniques, or which is a bias of being extremely selective in the population of EVs that you're isolating for your samples. And so this is something that must really be taken into account. And this is why, let's say, this field of investigation is extremely challenging. And in addition to this, I also want to bring another point, that basically, if you consider the different application that you can figure for EVs, ranging from, let's say, the analytics, microscale analytics for diagnostics, for example, but how you translate the information that you can get from EV analytics to other scale of separations, for example, for laboratory purpose, for, let's say, normal biobiological investigation of their function, which can bring them to another level, which could be their perspective view of some subpopulation as a therapy. But the thing is that across different scales, it is often the case that people uses different methodologies for separation, and this basically somehow makes it very difficult to compare the information that you can get across scales, across different settings, and so altogether it's very, very complex puzzle to be solved. And this is basically what is currently still frustrating the true translation application of EVs, especially in the analytical field. I will mostly focus my presentation on this part of the story, because basically how to isolate pure EV preparations is still a matter of debate, and I would say there is increasing awareness about the fact that there's no the best way to do it. It's always a kind of compromise, and this is truly related to the complexity of the VIA analytes themselves. And this can be true both for diagnostic workflows or just for QC protocols when you have to deal with let's say, EV preparation in the pharma industry, for example. And of course, this reverberates also on the poor standardization that you can have across different laboratories and different settings. So after a while that people really try to get the most pure EV fraction, now somehow someone is basically debating, is this really the way to go, or we should focus more about having robust and reproducibility methods that can allow real use of EVs in analytics, in biomarker discovery. And so basically, after, let's say, more than 10 years of intense efforts, now people are let's say, shifting a bit that kind of approach. But this doesn't mean that we have to compromise the quality of separation, the quality of analysis, just for being more reproducible and easy to use methodologies. And basically, if we, in this sense, new technologies are what are really needed and what are really helping us today. Because if you go back to the heterogeneity of EVs, Now we have increasing number of techniques that can allow us basically to sort the complex EV information that we have as a whole, and to be able to dissect the presence of different EV subpopulations, and to truly assign individual roles to each of these population, which is something that really is expected to make the difference in the future ahead of us. And basically we are experiencing in the EV analytics field a sort of shift, or at least we have two parallel worlds now that we are experiencing the shift from bulk analysis, traditional analysis like Western blot, ELISA, to technologies and techniques that allow us to really zoom in and be able to analyze more in detail the whole panorama of EV, or how EV components. And there are really a lot of techniques which are emerging, and all of them can give us different levels of information, from microscopy techniques that can tell us a bit more about their structure, about their nanomechanics in the case of AFM, for example, to those techniques which are supporting us more and more and providing us more and more efficient tools to analyze the composition and the biomarker profile which is associated to individual extracellular vesicles. And in this way, this is how we decided basically to operate. So as a group that is trying to basically to develop and to streamline the EV analysis, we wanted basically to use, let's say, the right technologies to be able to titrate single bio-nanoparticle populations, because in principle this will enable us basically to run direct analysis of minimally treated samples in this case. So we want to basically in our workflow to avoid pre-isolation of EVs, as this isolation step, as I showed you before, can really introduce artifacts bias, or can be really hard to standardize across different laboratories and settings. On the other side, we are looking for technologies that also allow us basically to be operated in a sort of clinical compliant workflows. So must be to some extent high throughput, must be easy to operate, and in other words, must be something that can be managed by, let's say, a trained lab technician, and not just like by super expert researcher in this area. On the other side, as I introduced in my first slide, we have two souls. One is, let's say, dedicated to technology. The other one is dedicated to chemistry and to molecular tools. And so in the second part of my presentation, I will show you how we try to address this problem related to the heterogeneity of EVs, developing new molecular tools which are dedicated to the study of EVs. But I will start from the first part. So a few years ago, we were in the position, okay, we are getting projects on EVs and how to make them, how to advance them towards real clinical scenario in the future, possibly, but to introduce robust protocols for analysis. And so we started to basically to compare the technology that we had available in the lab at that times. And in particular, we compared at the beginning two different technologies. We are to some extent complementary with each other, and while different to some other extents. For example, the ExoView platform is a microchip based platform, whether single molecular array technology is a bits based technology. But they share this point about giving us the possibility to digitally sort the signal, the analytical signal that we have, and I'm going too much more in detail right now. So here the idea and the purpose of our study was to pick which was the best, and not the best, of course, in an absolute way, but the best that could better fit our purposes and our future goals. So just a few words how these two technology works. As I said, the NanoView, the ExoView platform, as it was named a few years ago. Now they've been acquired by another company that changed the name, but the technology is pretty much similar. So basically, there are chip-based techniques where basically EVs can be captured on the microspots by the use of antibodies. And then you have two levels of detection. One is interferometric. So you have a first level of detection, which is based on the interferometric index, typical of biological nanoparticles. And then you can run a second step and fluorescence staining of potential surface markers on the surface of EVs to probe the presence and abundance of different kind of putative biomarkers. What is key here is that this instrument has a digital counting possibility, meaning that you do not read the whole area of your microspots, but you are reading single counts event, which then the instrument can assign as positive events, meaning that virtually you can really go down with sensitivity. To some extent, the Simoa, single molecular array technology, work on similar principles despite, let's say, totally different technology. I'm sure that anyone by Quanteris can give you a much better description than I will do right now. But still, what I want just to highlight how we can basically out of switch from a bulk signal to a more precise counting and better selectivity of events. In this case, the platform works on microbeads, magnetic microbeads, which are functionalized again with, let's say in this case, antibodies for the target analyte. And then upon implication, basically beads are individually sorted into these disk arrays, where basically beads are individually allocated in microwells. And then the instrument again can perform a digital counting, a digital assignment of on-off events that can basically translate in a much better sensitivity. And this is what we need to go back to my introduction. This is the kind of technology, the kind of principle that we need in order to analyze very complex samples when we are after very, say, when we want to go after subpopulations, and we want to basically to be able to analyze low abundant analytes and get much better resolution of our analytical signal. And basically what we did was to basically customize the two platforms with very similar principles. In one case was microchip, in the other case was Simoa platform, but we basically ran very specular assays, which were based on functionalizing the respective solid supports, so microchip and microbeads, with antibodies targeted against the most common and the most, basically the gold standard usually in immuno-based assays for extracellular vesicles, which are anti-CD9, CD63, and CD81, which are some of the most common EV markers associated to the surface. And then we started to establish some comparative assessment of the two technologies. And to cut a very long story short, we also benchmarked results with other, let's say, state-of-the-art and benchmarks, including super resolution microscopy. And what we could experience is that by the Simoa platform, we were able to get higher sensitivity and better dynamic range, and we could get a better correlation with other analytical benchmarks, which was very important to us at the beginning. Plus, to fit our purpose, we had also better high throughput possibilities for analysis. And so basically, despite the ExoView platform can still provide us some other relevant information which can be complementary in some basic studies about the EVs, we decided to stick for the moment with Simoa technology and push further other concepts towards validation. So basically the principle here, we have a right technological tool to go on, to move on our research and to see if we can really make additional steps forward. And this led up to another problem, which is the intrinsic heterogeneity of EVs. So if you have to devise an analytical immunosandwich for EVs, the first question that you ask yourself is, which antibody will I use to target EVs? Because again, extracellular vesicles composition is not stable. You have a lot of variety between different EVs, and especially you have a different abundance that can of some, even for the most typical markers of interest. Typically this expression is not stable. It can be very tricky. And this is just to show you on the left hand side how much the assessment of other additional clinically relevant markers can vary just if you switch the antibodies that they use for capturing EVs. And thus, of course, this has a massive implications towards real diagnostic scenario or real biomarker discovery scenarios, because if the analysis is so much influenced by the kind of let's say analytical probe that you use to fish for EVs. On the right hand side of the screen is basically exactly the same concept, but here is even more reinforced by the fact that even when you use a combination of different antibodies, and in this case was the 3 antibodies directed against the 3 most common EV markers, or the 3, let's say, gold standard EV markers, which are tetraspanins, you may end up losing up to the 40% still of the clinical value of other EV associated markers. And this again, of course, is something that is now emerging more and more due to the fact that now we have better and better tools to go deeper and to understand a bit more about individual composition of EVs. So basically, the principle, what we try to address was exactly this point, is whether we could basically switch the paradigm in how we capture EVs. So basically, we were trying to figure out a way to devise what we call universal EV binders, which are, of course cannot be universal, let's say, in its most broader sense, but new tools that could give us, let's say, better analytical possibility towards an unbiased EV binding. And then, of course, it was critical to us to have something in hands that could allow the integration on different platforms, ranging from analytical platforms, but also to the possibility to scale up this way of isolating EVs and just to recall what I mentioned before, basically to be able to act like a glue between different scales of operation and settings of operation. Basically, what we got inspired by the fact, which is for totally unrelated field I was investigating at that time, the process of membrane curvature sensing, which is physiological process that is basically very relevant to the physiology of cells, including intracellular organelles trafficking and membrane remodulation and many different things. What we were inspired from is that the fact that some proteins can really sense in a very selective way a biophysical property, which is membrane curvature. And the way they do that is basically usually these protein domains are amphipathic in nature, and they can basically insert within what are called lipid packing defects, which are due to the tension which is generated in highly curved membrane structures. But despite this field is very, let's say, is very young and super interesting from, let's say, a theoretical point of view, we realized that there was almost nothing about the possibility to exploit this phenomena for applied solution in research. And so basically our concept was, can we consider actually to target the curved membrane as a sort of universal epitope that is shared across all the different EVs populations, as they basically, most of the interest on the analytical side is currently focused on EVs ranging in this size, 50 to 200 nanometers range. And basically it's quite unique characteristic in the extracellular space, which also distinguish them, for example, for circulated lipoproteins. And so the idea is whether we could basically be inspired by these proteins able to sense the membrane curvature, transform their domains in synthetic peptides, and use them in the place of antibodies to target the membrane as a more pan-specific, universal EV epitope. And basically, the key concept here were that if we were able to provide this tool, we would have a tool, let's say, first of all, to have an unbiased binding, and then to be pan-specific and to most EV subtypes, and also across different, not only not strictly related to human EVs, but also to other species EVs, which is of course very critical when you're working with animal models and so on towards research. On the other side, as synthetic peptides basically can be produced quite easily and conveniently in a laboratory up to the gram scale, basically they can be extremely versatile, scalable, and you can really play with them and customize them depending on the application that you look after. So basically we started investigating some of these sequences which were present in the literature, realized them in the form of synthetic peptides, and we started testing them for their ability to basically to isolate and capture EVs. And so again, of course, I have no time probably to show all the experiments that we ran in the first place, and all the basically the screening work that we did to finally obtain what was the sequence that performed the best in our hands. But this is just to show you that basically we were able to basically to isolate EVs by these methods, to assign them let's say that to basically to confirm that the biophysical traits of the particles that we were isolating were the ones typical of EVs, that we could find all the markers there. And then a very specific features is on the panel on the bottom line, which is that when we compare the isolation of EVs from plasma samples, the depletion of EVs from sample sample, and we compare the use of our, let's say, universal peptide binder with the ones of individual antibodies, we could see that we have a much more consistent depletion when we check for with common EV markers. So basically, somehow we are not restricting our selection to some particular EV subpopulation, but we are able to collect all the message, all the circulating information that are out there. And so in recent years, basically we started to couple this molecular technology to Simoa technology to try to see if we could combine and synergize the two different tools. And basically we started doing very systematically by comparing what were in our hands the gold standard, which was the use of Simoa beads functionalized with a mixture of anti-tetraspanin antibodies that we call tetrabeads, and to compare basically the analytical performances that we could get with the ones that we would get by MSP peptides running exactly the same assays in parallel. And basically the results shown here are that we are pretty much doing well in this comparison, and then actually we can do even better in some cases if you compare the two graphs on the top. But what is actually even most important to us was that we could run this kind of analysis directly into clinical biospecimen like plasma and serum. And so we could actually totally skip the first step in EV analysis, which is often used, which is pre-isolation of EVs. So we go to direct analysis, which was our key goal in our assessment. Then we of course checked also for what are typically considered contaminants in EV analysis, which are basically the co-isolation of lipoproteins. And from the panel below, you can see basically that we have no difference between MSP functionalized beads and control beads, which make us, while we have a very strong signal which is related to the presence of EV markers. And so basically this was already basically all of setup work that we needed to move on and go deeper and deeper. The next step for us was to assess whether is actually basically MSP analysis reliable and representative. And basically what we did was to design comparative experiments again to probe how which say putative biomarker distribution we will get by using one approach or another one. And what we could see is that basically we get pretty much exactly the same biomarker profiling, whether in the case we use MSP or whether we use a mixture of antibodies that are then probed with the very same antibodies individually. Meaning that basically we get the same distribution, and this tells us that we are not losing information when we use MSP, and so that we can truly compare for specific cases MSP technology with the use of antibodies. Then there's another thing, which is basically EVs can have different biogenesis pathways. One is, and depending on the biogenesis pathway, they can in some cases totally lack the presence of what are most commonly used markers of reference, which are tetraspanins again. And in this case, we were provided by a group of collaborators some red blood cells derived EVs as a model of ectosomes, which are basically EVs which are budded from the plasma membrane directly without going through endocytosis pathway. And we used them as a model for two different things. The first one, again, was to probe that we were able to analyze still EVs totally lacking the traditional tetraspanin profile, which we succeeded. But also the interesting thing is that there is really a lot of people investigating the use of what are called EV analogs and EV mimetics, which are some kind of semi-artificial EVs, which again nowadays totally lack the basically the toolbox, the analytical toolbox to support the development, if you would refer to traditional use of antibodies. And so basically this kind of enlarge our field of application in the case of EVs. Finally, we also wanted to basically to try to understand if we can really also go and test this workflow in a real, say, in a context that really mimic a clinical workflow. And we did this in collaboration with a group of Lucio Barile at Cardiocentro Ticino in Switzerland, where Lucio is very active in the field of EVs in the cardiac injury. And so basically we were able to stratify patients experiencing myocardial infarction versus patients with stable angina. And we did this by a combination of different EV-associated markers, and the aggregate curves basically returned us a diagnostic power which was not inferior to the one of the gold standard, the clinical standard, which is high-sensitivity troponin. But most importantly, again, for us was the proof of concept that we could apply this technology and this workflow into a clinical viable way. Another level of information for us was that when we ran MSP-based analysis, we were also able to restore somehow the informative value of some markers, which was otherwise overlooked when traditional antibody-based approaches were used. And this brings us to basically to how we now feel what we feel we have now in hands. So we feel we have a very versatile tool towards EV analysis. So starting from the very same, basically chemistry, the very same ligand, we can probe different EV sources, human, but also non-human. We can probe with the very same technology, different markers that can be probed individually and apply them to very different contexts. And we have indeed we have basically ongoing testing from this very same approach that I just described from for the myocardial infection work. We have now tests ongoing for breast cancer, prostate cancer, neurodegenerative conditions, heart transplant. But also we are also testing these methods in the QC of organotropic EV analogs. So back again to that field of EV mimetics, which is emerging. So this is in principle to show you how versatile we think now we are in this moment in terms of the analytical possibilities, and we see this as an approach to streamline the biomarker discovery pathway of EVs. But then I want to take a little step back and stress the fact that we are using a synthetic peptides, and synthetic peptides again can be scaled up. So basically this open up the possibilities to bring immunoaffinity like isolation to other fields of application which are just not analytic one. So potentially to lab scale isolation, or even to manufacturing scale isolation, which is basically was the focus of our a European project which we called Marvel, and which was basically devoted to showcase all the possible versatile application of MSP technology. And the principle here is that, again, by having a synthetic tool that you can diversify, you can basically tailor its way of conjugation to many different supports, many different tools, and basically find your application in a sort of a plug-and-play solution. And we did show this by with the number of possible with a number of different collaborators and collaborations, and which I just report some of them. And what I want to highlight here is that actually only 2 of these work were run by us directly. The other ones were just people asking us to provide them the molecule with a suitable tag for their applications. And then, of course, it's always nice when something works in your hand, but it's even better where something works in other people's hand without too many difficulties. And that's exactly what we could show here, and what we are trying to expand right now in our range of collaboration. As basically, we're quite happy that our concepts are being increasingly appreciated by a number of people, and again, in many different contexts and scenarios, which makes us confident that we have a quite versatile tool in our hands. And this brings me also to very sad part of the story, as basically most of these efforts and developments did see my former colleague Marina, which really was the key center of this technology and this range of collaboration, which suddenly left us very recently. And a nice obituary paper recently appeared on JEV. I suggest you to have a look. And this slide is really to express my gratitude and for the ride that we have together, and to give her credit about this all of these developments and fantastic collaboration that we were able to set up. And it wouldn't be possible without her, and really we miss her as a person much more than a scientist, really, and she was exceptional. And this also brings me to acknowledge all the fundings and all the people in the group which are basically can make the magic happens every day. And of course I want to thank all the ones involved in the organization of this event. So Sarah, Byron, Mark, thank you very much, the guys from Quanterix, And of course, I'm really happy to take any question and answer any of your curiosity right now. Thank you.
All right. Thank you very much, Alessandro, for that. We're going to start the Q&A in just a moment. Before we do, a couple of notes for the audience. You can find some handouts on the right-hand side of your screen. There's a couple of things in there, so please do take a look and download those before you leave the webinar today. We're also going to send out, there's a couple of notes about the recording. We're going to send out the recording within the next 24 hours. So if you did join us late or you've missed anything, you can watch back anything that you've missed when we send that email. We'll be with you in the next 24 hours. All right, Alessandro, let's get to it. We've got plenty to get through. Let's start with this one. A lot of interest resides in the analysis of EB luminal markers. Have you planned any dedicated workflow in this direction?
Yes. Basically, in the results that I show you, we mostly focused on how to set up a streamlined, let's say, workflow for the analysis of external EB biomarkers, which are on the membrane. We are now trying to work on similar principle using the very same supports, basically microbeads-based tools and MSP ligands to pre-isolate EVs, lysis protocols, and analysis of internal markers. In our, let's say, most, in wishful thinking, we believe there might be an option also to basically even to avoid, let's say, pre-isolation followed by lysis, but to introduce a sort of detection step with the secondary antibodies and playing with the buffers and use of detergents, be able basically to do exactly the same immunosandwich scheme, but be able to look internally. Of course, this is to be tested, to be developed, but it's likely going to be one of the major aspects of our near future research. Yeah.
Fantastic. What about this one here? It's asking, do you envisage the use of peptides also as molecular probes targeting specific protein markers? If so, what could be the advantages of this?
I can say yes. Maybe here I'm a bit biased by the fact that, as I said, I spent basically my entire career working on peptides. What I can tell you is that is definitely in our pipeline to develop peptide-specific target-specific peptides. And the reason we want to do that is, of course, we may not be able to fully compete with the affinities that antibodies sometimes can reach. Still, now peptide technologies can allow you to develop peptide ligands which are quite good, meaning that it's not so uncommon to be able to reach affinity levels down to the low nanomolar range. And so in principle, the idea here is to replace the use of antibodies by synthetic ligands. We can share exactly the same principle of scalable immunoaffinity-based ligands, which can basically open up new possibilities, not only related to microanalytics or analytics in general, but also they could enable in the future to scale up the immunoaffinity capturing technologies in order, again, to try to merge the level of information that you get from the analytic with other working settings and scales of operation. So this is the main principle. Besides the fact that peptides might have additional features like being very cheap to produce, much more stable than antibodies. They can be shipped and stored in totally different conditions with respect to antibodies. So these are, let's say, extra advantages which are very practical but very relevant to everyday research. So this could be other possible reason to go into this direction is something that we will do absolutely.
All right, let's take this next one from Grace. She's asking, could you please share some insights on EV in neurodevelopmental disease, such as relative abundance, comparing with those from other tissue types?
Yeah, this is a very good question. It's a very debated topic. because of course neuroscience and tumors, I would say, that are those kind of areas where efforts are the most intense. And basically the thing is that in the neuro area, of course, the ideal goal is to be able basically to isolate circulating neuro EVs which are coming from the brain directly into the blood, especially if associated with condition where you might have, let's say, an injury to the blood-brain barrier, which could facilitate the penetration in the bloodstream of neural markers. Here is this field is pretty much debated because there's really a very intense effort going on about trying to understand which are the true markers which are really exclusive of neuro-derived EVs, and as a consequence try to develop here very specific, let's say, molecular ligands which can enable the enrichment of these specific class of EVs. And so this is kind of basically, let's say, for some reason, like opposite to what I just presented. So it's just another approach. So be very specific about a marker and try to find the right market which could enable the isolation of very tissue-specific EVs or organ-specific EVs. I know I'm not focused, let's say, so far in this area. New collaborations are about to start in this area for us, but absolutely this is something which is very intensively investigated. I think that over the next few years we will have a lot more than we have right now in this area.
All right,
I hope I've answered the question.
If not, Grace, then please do follow up and we
exactly.
send that through. This next one, Alessandro is asking, does the system work equally in plasma and serum?
Not really. We always found, I think it's more general question here, as we always found that we get better results when we are dealing with plasma with respect to serum for some reason, which is still not fully understood. Of course, it might be related to the clotting process that is involved. But in principle, even when we do use sometimes, I mean, antibodies for EV analysis, I don't want to give the impression that we totally eradicating the use of antibodies. We do see these peptides as a kind of complementary approach that add, let's say, different layers of information with respect to antibodies. But in both cases, we do actually feel plasma is much better sample to work with, more rich in EVs, better signals, and overall, it's more easy to use, at least in our hands with our protocols.
All right, let's take this next one. It's number 12 on the list. They're asking, in your workflow, you mentioned that you were able to avoid isolations of plasma proteins and lipoproteins. Does this include the exclusion of plasma, LGGs, and albumin, etc.?
Yeah, I mean, we of course haven't tested all the possible proteins. What I can tell you is that we definitely checked for the most abundant ones, and especially albumin was included. And we did check especially for lipoproteins, as again, we are also working on scaling up this technology for, let's say, isolation tools. And typically lipoproteins are, let's say, the most tricky contaminants to be removed. Of course, I mean, when it gets to only analysis, at some point, some contaminants may be still bound to the surface, to the bits, even in a specific way, and not related to the probe itself, but to the support. And here, basically, as a group, we also have internal workflows where we basically we work on coatings for the surfaces of many kinds to serve both as tools to basically to drive the mobilization of ligands, but also to prevent fouling of circulating contaminants and proteins. Probably it's another field that we will expand soon, and probably we will go through more systematic assessment of possible contaminants, let's say, covering more proteins than we did to date, basically.
How about this one? Paul's asking, I wanted to ask you if Alessandro has ever looked at saliva specimens. He says saliva is a very rich source of EVs, and we have developed a very simple technology to collect EVs simply in 1 to 3 minutes, even in home-based situations. Would you be interested in potential collaborations? It's number 10
absolutely.
on the list.
Yeah, I mean, the message is actually for everyone, because again, we are actually, this is not a joke, because we are, of course, at a stage where we really appreciate feedbacks from other users of what we developed, and so totally, I would say that the saliva is probably the only sample which we've never worked with so far. We know it's really interesting and very very kind of hot kind of samples towards future home-based diagnostics, etc. So definitely we'll be happy to collaborate on this area as well.
The next one's asking, is it possible to get an absolute quantification with the Simoa?
Yeah, this is this is let's say, I would say this is a very good question, and but this is more general to the entire EV analytics field, because of course when you deal with such a heterogeneous analyte in general, of course a key question could be what could be a good reference. Usually when you set up an immunoassay for a specific protein marker, you can just buy that specific marker, do a titration curve, and basically be able to quantify, to build calibration curves, etc. In the EV area, this is not so trivial. There are many commercial standards which can be used for this purpose, but of course maybe they are from cellular origin, and so they have, let's say, a precise markers distribution which is typical of that cells of origin. On the other side, I know there are out there many synthetic standards, meaning they can be liposome functionalized with protein markers on the surface, which can basically in principle allow you to have a more level of control about the reproducibility of the source, and possibly be able to to truly quantify the signal which is associated to your marker. I would say that in this area, probably there's still not a gold standard, and it's something that we are working on our side as well, because the idea is even before going to absolute quantification, I would say that the most urgent matter is how to correlate results from different laboratories, even using the very same technique. Because sometimes, just like an installed instrument in one place could work slightly different than in another one, or just the very same sample, due to traveling, shipping, could suffer from a slight change in the signal. And you need some kind of a tool, a standard to be able to equalize results. I would say that this is the most needed thing right now. And then the next step will be to move towards really quantitative analysis, meaning that something that where we might express results in terms of absolute protein content related to some EV specific marker, at least in my opinion, of course.
Superb. Well, we'll take a few more, Alessandro. We are getting quite close to the hour, but we'll try to get through as many as we can. Yeah. A reminder, if we haven't answered your question, if Alessandro hasn't answered your question, we'll pass them over to him. He can come back to you. This next one starts off by saying thanks for the great talk, and there's been a lot of positive feedback, so that's always good to see. Can you elute the captured EVs from the peptide functionalized beads? It's number 15 on the list, Alessandro.
Yep. We devised some preliminary protocols, I would say, for release. And basically here the thing is that we are trying to do it in a very, say, most mild possible conditions. And we do this by increasing some salt concentration, especially we tested so far divalent cations-based buffers. And here is again a matter of investigation that we have to go much deeper right now. We focus, say, we really pushed in the last year or so towards this analytic tool, but now it's time for probably to address this point more, again, systematically. The principle here is that what I believe is that the interaction of the peptide with the membrane might be is actually, I think, individually, if you take the individual molecule, is probably weaker than usually an antibody target is, which is usually much stronger interaction. You have to increase a lot the salt's concentration, and this could be good or not, depending on the analysis that you have to run after that process. We have a feeling that the peptide membrane interaction is slightly weaker. Of course, we take advantage of the cooperative binding, meaning that if we have a surface covered with peptides, then we have multiple ligands which can somehow have an avidity, affinity. In our case, I feel that avidity is a very strong component towards the membrane affinity. This is to say that by playing with buffers, and we already have actually data in-house that show that we can reduce vesicles, we probably might need to optimize and be more quantitative about how we're good at reducing them.
Superb. We've just ticked over the hour, but let's take one more question before we wrap things up. This one from Arthur's asking, have you explored the affinity and potential use of MSPs for studying bacterial extracellular vesicles, such as those derived from the microbiota?
Not those from the microbiota, but I have a running grant on bacterial leaves from Mycobacteria tuberculosis in collaboration with the University of Milan. We have preliminary data that show that we can still have binding. Not entirely sure whether we are really performing the same as in this case, as let's say bacterial leaves has also additional level of complexity. I would say in principle, and it's also some other groups which follow up with this, say, very similar approach using peptides for membrane recognition. There's something published already which deals with the possible use of membrane targeting peptides for bacterial disease analysis. So that's something that probably we want to expand ourselves in the future. Yeah.
Superb. We'll wrap things up there. Alessandro, thank you very much for sharing your expertise with everyone today. Big thank you to everyone in the audience for joining us. If your question hasn't been covered, like I said, we'll hand them over to Alessandro and the team and they can follow up with you. A final reminder about the handouts. There's a couple in there on the left-hand side of your screen. Please do download those before you leave. And you'll also see a survey appear on your screen once you leave today. So if you could share your feedback with us, that would be excellent. Alessandro, any final thoughts before we finish up?
Oh, no, just again, I'm totally available for anyone to reach out and just ask more or want to know more, or maybe try if we can set up new collaboration. Definitely, we are always try to be open to anything and receive any kind of feedbacks, even if negative, that will be is still part of the job. And still let me thank you again, Quanterix, for this invitation, and it was very good, and I'm very honored to be able to present our work here today.
Superb. Well, thank you again, Alessandro, and thank you again to everyone for joining. We wish you all a pleasant rest of the day, and hopefully we'll see you all again very soon. Thank you very much.